You are born inside a hive that most people would never approach twice. You look almost identical to the honeybee your neighbor keeps in a backyard garden. Your body is the same size. Your wings are the same shape. Your sting carries the same venom in the same amount. But something inside you is fundamentally different in a way that cannot be seen from the outside, felt from a distance, or measured by looking at you resting on a flower. Something that has made your species responsible for hundreds of human deaths, the collapse of livestock operations across multiple countries, and one of the most consequential accidental scientific experiments in the history of modern biology. You are an Africanized honeybee, and this is the story of how something built to survive became one of the most feared insects on Earth, not because of what it is, but because of where it ended up. Cornell University has studied Africanized honeybee populations for decades, mapping how a single escaped subspecies spread from a research facility in São Paulo, Brazil, across two continents in under 40 years. The Smithsonian Institution describes this spread as one of the most dramatic biological invasions ever recorded in the Western Hemisphere, a living case study in what happens when the wrong organism reaches the wrong environment at the wrong scale. You hatch in a wax cell inside a colony that operates, at first glance, exactly like any honeybee colony in the world. You emerge soft and pale from your cell. Within hours, you begin cleaning chambers. Within days, your glands begin producing food for larvae younger than yourself. You cycle through the same age-based roles every honeybee worker follows: cleaning, nursing, building wax, guarding the entrance, and eventually foraging. Every stage of your development mirrors the life of a European honeybee so precisely that trained beekeepers in the early years of your species' spread in South America often failed to identify the difference until the moment of disturbance, the moment that changed everything. Because the difference between you and a European honeybee is not in your body. It is not in your venom. It is not in your honey production, your foraging patterns, or your loyalty to your queen. The difference is in how you respond when something threatens what you have built. Entomologists call it intensified defensive behavior. What it looks like in practice is something far more alarming to anyone who has witnessed it. Where a European honeybee colony might send 50 to 100 defenders to investigate a disturbance near the hive, your colony sends thousands, sometimes tens of thousands, within seconds of the first alarm pheromone signal reaching the air. And unlike a European colony, which typically breaks off pursuit after 30 or 40 meters, your colony pursues a perceived threat for distances that can exceed 400 meters, continuing the attack relentlessly long after most animals, and most humans, have reached what they expected to be safety. Is this aggression in the way humans mean it? Or simply survival logic, refined across millions of years in an environment where threats were bigger, faster, more persistent, and more determined than anything the honeybees of Europe ever needed to prepare themselves for? The Royal Entomological Society has documented exactly how Africanized honeybees evolved their extreme defensive capability. Your ancestors lived on the savannas and forests of sub-Saharan Africa, sharing territory with honey badgers, which are among the most relentless nest raiders in the natural world, large primates with hands capable of tearing open a hive, and human hunters who had been harvesting wild honey for thousands of years using smoke and fire. In that environment, a colony that hesitated when threatened did not survive long enough to pass its genes forward. A colony that responded with overwhelming force, immediately at massive scale, before the threat could reach the core of the nest, that colony survived. That colony reproduced. That colony's defensive trait became the norm. You were not bred to be dangerous. You were shaped by a continent that required intensity, and that shaping happened across hundreds of thousands of years before anyone understood what it would mean to move you somewhere else entirely. Let's reconstruct how that move happened, because the history of the killer bee is inseparable from a single decision made in 1956 in a research station outside São Paulo. A Brazilian geneticist named Warwick Kerr had spent years studying honeybee subspecies across Africa, looking for traits that might make bees better suited to South America's tropical climate. The European bees already established in Brazil were struggling. They produced modest amounts of honey. They were poorly adapted to heat. They resisted disease inconsistently. The subtropical regions of Brazil, in theory, should have been ideal for large-scale honey production, but European subspecies were not performing the way researchers hoped. Kerr identified Apis mellifera scutellata, a honeybee subspecies native to eastern and southern Africa, as a strong candidate for improvement. This subspecies foraged more aggressively, worked longer hours during the day, demonstrated stronger disease resistance, and thrived in warm climates. The plan was to breed selected African queens with European drones to create hybrid colonies combining the best traits of both. 26 queen bees were imported. Colonies were established at the research station with queen excluders, metal screens that prevent queens from leaving the hive to swarm, placed at hive entrances as a standard safety measure. In 1957, a visiting beekeeper removed those excluders, possibly not understanding their purpose, and 26 African queen-led swarms escaped into the Brazilian countryside before anyone could stop them. The consequences of those 26 swarms are still unfolding today. Nature has published multiple analyses of the Africanized bee expansion, tracking how colonies move through South America at rates of up to 500 kilometers per year in some regions, interbreeding with European bee populations along the way, but consistently maintaining the extreme defensive and reproductive traits of the original African subspecies, which proved genetically dominant in nearly every cross. By the 1970s, killer bees were established across much of Brazil and spreading into neighboring countries. By the 1980s, they had reached Central America. By 1990, the first confirmed Africanized colony was identified in the United States near Hidalgo, Texas, completing a journey of over 10,000 kilometers that had taken 33 years and had never been intended to begin. Today, Africanized honeybees are established across the southern United States from California to Florida, throughout all of Central America, and across almost the entire South American continent. They are the dominant honeybee subspecies in most of the regions they have entered, having largely displaced European bee populations in tropical and subtropical zones. This is the counterintuitive twist that surprises almost everyone who learns the actual science behind the fear. Africanized honeybees are not more venomous than European honeybees. A single sting from a killer bee delivers no more venom than a single sting from a backyard hive. The venom itself is chemically identical. The danger is not in the sting you receive. The danger is in how many stings arrive at the same moment. The University of Exeter has studied large-scale defensive responses across bee subspecies, documenting how the physiological threshold for serious harm from bee venom is typically reached somewhere between 500 and 1,500 stings in an average adult human, depending on body weight, sensitivity, and speed of response. European honeybee colonies, even when significantly disturbed, rarely deliver more than a few hundred stings in a single encounter. An Africanized colony defending its nest can deliver that number in under 60 seconds and sustain the attack for far longer. A human who cannot run, who is caught in an enclosed space, who trips and falls, who has an existing sensitivity to venom, or who is very young or very old faces a genuinely life-threatening situation in a way that a European bee encounter almost never creates under normal circumstances. For livestock, the risk is even more severe. Cattle, horses, and pigs cannot outrun a pursuing swarm in an open field. They cannot shelter effectively. They cannot communicate that they need to move away from a hive they may have unknowingly disturbed. Documented cases of livestock deaths from Africanized bee attacks extend into the thousands across South and Central America and represented a serious economic impact on farming communities in affected regions during the decades following the original spread. Beekeepers managing Africanized colonies must adjust every aspect of their practice. Heavier protective suits, more smoke, applied more carefully and at greater distances before opening a hive. Slower movements, more predictable schedules, since Africanized colonies respond more intensely to irregular disturbance. And constant awareness that the margin for error in management is significantly smaller than anything European bee management requires. Let's reconstruct a day inside your colony to understand why that margin is so narrow. Your alarm pheromone system is the core of your colony's defensive advantage. The chemical responsible is isoamyl acetate, a compound released when a bee stings or senses threat. It smells faintly of bananas to a trained nose. To your sisters, it is an immediate and absolute call to mass mobilization. The moment a guard bee detects a threat near the entrance of your hive, she releases this pheromone. The first wave of defenders emerges within seconds, targeting the source of disturbance directly. When those defenders sting, they release more isomyl acetate, amplifying the signal. More bees emerge. The signal intensifies. Within 2 minutes, a full defensive response can involve thousands of individuals, all targeting the same location, all releasing additional alarm pheromone with each sting. European bees use the same system. The threshold for triggering it in your colony is simply far lower, the response far faster, the escalation far more complete. Researchers at the Entomological Society of America have documented how Africanized colonies respond to disturbance stimuli that European colonies simply ignore. Sounds, vibrations, dark colors moving near the hive, or the scent of carbon dioxide from breathing at close range. Your colony is tuned to react. And once reacting, it does not easily stop. Here is what surprises most people who study you closely. For all the fear your defensive behavior creates, you are in many ways a more effective bee than your European counterparts in the environments you now occupy. Your colonies swarm more frequently than European colonies, sometimes every few months, which helps naturally reduce varroa mite loads by interrupting the mite's reproductive cycle. Varroa destructor, the parasitic mite responsible for widespread colony collapse in European bee populations around the world, has devastated managed beekeeping operations on nearly every continent. Your colony's higher swarming frequency is one reason Africanized bee populations have shown greater resistance to varroa damage, with some colonies demonstrating hygienic behavior that removes infested brood before mites can fully reproduce. You forage earlier in the morning and later in the evening than European bees, extending the productive hours of each day. Your colony responds more quickly to nectar flows, building up population faster when resources are available, and swarming to exploit new areas before European colonies have fully mobilized. National Geographic has reported that in Brazil, honey production actually increased significantly in many regions after Africanized bees became established, because your foraging efficiency outpaced what European colonies had delivered in those same environments, even as the management challenges for beekeepers intensified dramatically. You are not a failed experiment. You are a highly successful bee placed in a context that was not designed to receive you, functioning exactly as your environment shaped you to function, with results that are complicated, costly, and in some ways genuinely impressive. Does that dynamic feel familiar? Many of the most consequential disruptions in human history were not caused by things that were broken. They were caused by things that worked extremely well, just somewhere unexpected. A technology, an idea, a species, a policy, something optimized for one environment, transported into another with outcomes nobody fully anticipated. The dandelion is the perfect lawn plant until it arrives in your garden. The cane toad is a perfect insect predator until it is placed in Australia. Warwick Kerr's African queens were perfect bees for the tropics until they left the research station. The story of your kind is still being written. Geneticists are studying whether the extreme defensive trait can be selectively reduced through careful breeding programs without losing the foraging efficiency and disease resistance that makes your genetics valuable. Some researchers believe a middle path is possible, a hybrid subspecies that combines African vitality with European manageability, one that has not yet been reliably achieved at scale. Meanwhile, researchers at Cornell and the University of Arizona are tracking how rising temperatures in North America may allow your populations to expand further north than previously predicted, moving into regions where European bee populations currently remain dominant. Your colony continues doing what every bee colony has always done. Queens lay eggs, workers build wax cells, foragers carry nectar and pollen back to a hive that hums with the same ancient rhythm it has always kept. Young bees clean chambers they were born in. Old bees fly until their wings give out. Somewhere nearby, a scout is evaluating a new location, a hollow tree, a wall cavity, a gap in a fence post, or a forgotten water meter box, assessing whether it is suitable for a new colony that will start the whole cycle again. That scout has no awareness of the history she carries, no memory of a research station in Brazil, no understanding of what her species has meant to the farmers, the livestock keepers, the hikers, and the scientists who have spent 60 years trying to understand her. She simply lands. She investigates. She returns to the swarm to report. You are born inside a hive that most people would never approach twice, and somewhere nearby, a new hive is already forming, built by the same intensity that makes you extraordinary, and the same intensity that makes you dangerous. The two things inseparable, the way they have always been, the way they were always going to be.